Hi everybody, welcome back to another video. This one's going to be all about translation or protein synthesis. So we're going to go ahead and just jump right in. Now there are a couple of things that you're going to want to know when it comes to translation. The first being that it proceeds through three phases, which are initiation, elongation, and termination. Now that might sound very familiar, like a start, middle, and end, because it is like a start, middle, and end. Initiation means basically to start. Elongation is not quite middle, but it does mean that we're elongating the polypeptide during that time. And termination is much like the end. You're also going to want to be aware of the three types of RNA and their roles throughout translation. So understand that there's a lot of complexity that we don't go into detail, and there's also some complexity that we do go into detail, but always have these two bullet points in the back of your mind to really guide how much you need to understand. So we're going to go ahead and start with some of the differences between prokaryotic translation and eukaryotic translation. And if we're actually talking about what's happening at the ribosome, it's not all that different. But to get to the ribosome, it is. So recall, in eukaryotes, we have DNA that's in a nucleus that is being used as a template to make mRNA. But even once we have that mRNA, there's a lot more steps that are going to happen before it actually even leaves the nucleus to be translated. That's RNA processing. Whereas in a prokaryote, you don't have that nucleus barrier. And so as soon as transcription occurs, that mRNA molecule is now ready to be translated by the ribosomes, and that's what happens. So there's no RNA processing, and we say that transcription and translation happen simultaneously. And so it also happens usually a lot more quickly. And so that is something you should also be keeping in mind. Now we are going to be talking about mature mRNA, and when I say that, you should understand that it's a eukaryote. Once we get to the ribosome, it's pretty much applicable to both. But just keep in mind these differences because it does become very important, especially for some FRQs that you might see. So let's begin our discussion by talking about the role of mRNA. mRNA serves as the message that is decoded by the ribosome. The M stands for messenger, so it makes sense that it's going to serve as a message decoded by the ribosome. So how does this happen? Well, we are going to split that code into something called codons. And codons are just triplet codes of nucleotides. So we read 3 nucleotides at a time. That counts as 1 codon. And it turns out that 3 nucleotides coding for 1 codon in turn codes for 1 amino acid. So if we look at this picture on the bottom here, we see 3 nucleotides, an A, a U, and a G. That is 1 codon, which codes for 1 amino acid, which is methionine. AUG is always my start codon, and it always codes for methionine. Then we have ACG. This is a second codon. In this case, it is going to code for threonine. And you're going to read the entire mRNA molecule just like that, and 3 at a time, which will code for 1 amino acid. Now, there are 20 amino acids that make up proteins, and this is actually part of the reason why people used to think that protein might be the genetic code. We have since proven that that is not the case, but because there are significantly more amino acids than there are nucleotides, the thought was that was the only way to get such a high level of diversity in life. Now, you are not responsible for memorizing the names of amino acids. You are not responsible for memorizing the 3-letter abbreviations or the 1-letter abbreviations, but I do kind of want you to be comfortable with some of these amino acids, and especially in how they end. Most of them are going to end in INE. Ene, alanine, arginine, asparagine, cysteine, glutamine, all of them are ending in E. So if you see that, or you see other words like creatine, creatine is not an amino acid, but it is from an amino acid base, you might be able to make some assumptions about that molecule. If you do the math, you should have probably figured out that with 4 different bases and 3 different slots to put those bases in for each codon, then you should get a total of 64 different codes that you could get to represent 20 amino acids. That means that there are going to be many amino acids that have multiple codes that code for it. So alanine has 4 different codes that code for alanine. For arginine, there are 6 different codes that code for arginine. So the ones that have multiple codes, these are degenerate. If they only have 1 code, such as methionine or tryptophan, then we call them unambiguous. And this is really just a matter of the math here. Again, if we have 3 different slots to put 4 different types of bases, that's going to be 4 to the 3rd, which is 64 different potential codons. So we say that the genetic code is redundant because we have so many extra codons to fill 20 amino acids. We also say that the genetic code is universal. Now, in class, I gave you an example of potentially an exception to the start codon, which exists in E. coli, for example. That is way beyond this course. It's just something that I find really interesting from a purely scientific inquiry standpoint. But the fact that genetic code is essentially universal is actually incredibly useful in a lab. And almost every single person that I teach has somebody in their life that benefits from this directly through the advent of insulin synthesized by bacteria. So how does this work? Well, humans produce a gene for insulin because insulin is a protein, so there must be a gene that codes for the mRNA, which will then code for the protein insulin. And if we can isolate that gene, and we will talk about some methods later, but for right now, just understand that we can isolate an insulin gene, and then we provide it to bacteria. Well, bacteria will sometimes take up that gene through the process of bacterial transformation. And if they take up that gene and add it to their plasmid, now we have this thing called a recombinant plasmid. We say recombinant because it includes the insulin gene along with the bacterial genes in the plasmid. So it is recombinant. Well, if they have now taken up this gene, they will likely express this gene. And if they express the gene, then now we have the protein, and then we can extract it. Now, there's a little bit more steps that I'm not including here, right? You might link it with a reporter gene, which we will get to later on, or I'll talk about it in class, but I'll say it's beyond the scope of the course. But understand that this only works because the code is universal. If for some reason bacteria use a different set of codons to code for each amino acid, meaning maybe the start codon is not T, it's RNA. Let's say that it is CAG and arginine is CUG instead of what we saw on the previous slide, then it wouldn't make the same protein. But because essentially all organisms use the same codon chart, we can use bacterial vectors to make proteins that are not native to that bacteria. And that ends up being super useful to us. In fact, a huge percentage of our population who is afflicted with diabetes uses bacteria-grown insulin. And I think that's pretty cool. Now, we talked about mRNA. mRNA serves as the message. Now let's talk about rRNA, which is the major player at the ribosome. So what is the ribosome? Well, you probably learned it was the site of protein synthesis, and you're right. But it's also a large, basically protein complex that is interlaced with rRNA. So there are two components, rRNA and protein. They make up the two subunits, the large and the small subunit. Now, if we look at a good picture of this, then you will see that rRNA, here colored in orange, and proteins in blue, it really shows that rRNA is the major player. Now, what do I mean by that? I mean, it's probably the more important piece. And it is, because the rRNA is the thing that is actually catalyzing peptide bond formation. Now, that might sound a little crazy to you because you hear this term catalyze, you're thinking an enzyme, and enzymes are proteins. And you're right. The vast majority of things that catalyze in a cell are enzymes. rRNA is an exception. rRNA is a nucleic acid that can act as an enzyme, and it does. It is going to catalyze the formation of the peptide bond between two adjacent amino acids. Now, I included this image over here next to the image I just talked about. This is from Leishmania tarentulae. It is a parasite. This is the mitochondrial ribosome. Now, in this case, actually, the orange and the blue here are the protein, and the yellow is the RNA. And I show this because there's a lot of variability. So generally speaking, the rRNA is the larger component, and it just uses these proteins sort of as the backbone, the scaffold that it builds off of. But you can have a huge amount of variation here. Leishmania tarentulae, this RNA is actually the smallest that I know has been discovered, but it's worth noting that there is some variation here. Now, that's the role of rRNA. It's catalyzing peptide bond formation. Now, tRNA is what delivers the appropriate amino acid to the ribosome for polypeptide elongation. So this is the step that you need to know the best, elongation. So what exactly is tRNA? Well, it's transfer RNA. And that means that it transfers the amino acids to the ribosome. And it does it through this mechanism where it matches an anticodon to a codon. So what exactly is the anticodon? Well, it is going to be the complementary base pairs to the codon. So the anticodon is 3 bases or 3 nucleotides that is going to be opposite to the codon. In this case, it is U, A, and C. Now, that means then that it is the anticodon that is going to dictate which amino acid to bring. So, in this case, UAC anticodon brings methionine, and because UAC always is going to be complementary to AUG, we see that AUG codes for methionine. So this is how the correct amino acid is brought based off of the codon. But how does the actual amino acid get added to the tRNA? Well, in that case, we're going to rely on an enzyme. And that enzyme is called aminoacyl tRNA synthetase. So this is an enzyme that charges the tRNA with the correct amino acid. When I say charge, I don't mean that it is adding a positive or a negative charge. I really mean that it's loading the amino acid onto the tRNA. And it does it through 2 steps. It's going to phosphorylate an amino acid, and then it's going to transfer that to a tRNA. So if you watch this animation a couple of times, I think you'll see exactly what's happening here. I'm going to let it play through a couple times and describe what is going on. So, like I said, the very first step is going to be the amino acid is phosphorylated by ATP. So aminoacyl tRNA takes in valine in this case. It's going to take an ATP molecule and phosphorylate that valine. Then the appropriate tRNA comes in and is going to add that amino acid to the tRNA. Notice that this takes energy. And you're going to see in a minute that every single amino acid is brought by a tRNA. So if you have a polypeptide that is several hundred or even several thousand amino acids long, you have a lot of energy that you have just spent. That is very important to remind yourself of when we start talking about regulation. So let's go ahead and look at actually how this is happening at the ribosome. So I have a loaded tRNA, and that loaded tRNA is going to come in at the A site. What is sitting at the P site is the polypeptide chain. So the polypeptide chain is going to be growing from the P site, and as the ribosome slides down the mRNA molecule, that polypeptide chain is going to be added to the amino acid that once was in the amino site, the A site, but then is actually getting transferred to the P site in the process. I think it is really helpful here to watch this animation several times because this is really going to be how you're going to see what is happening in terms of elongating the polypeptide chain. So I will point out some things while it is playing just so you're super clear. This middle is the P site. This right side is the A site. The left side is the E site. So notice in this case, we are already partly elongating. So we have already a chain in the P site. Amino acid is going to come in, or tRNA with the amino acid is going to come in the A site. And as the ribosome slides down the mRNA, it is getting transferred to that previous amino acid. And also notice that every time that the tRNA is exiting, it's naked. It no longer has that amino acid anymore. It's going to go back and get recharged by that aminoacyl tRNA synthetase and come back once it's needed again. So I would recommend silencing me, put me on pause, replay this, and just watch through and describe it on your own to ensure you understand what's going on. Okay. Now I also want you to notice some patterns with these redundant codons. So if we look at alanine here, the first base seems to always be a G. The second base always seems to be a C. The third base could be an A, C, G, or U. If we look at glycine, we can see a G, and then the third base could be an A, C, G, or U. So what does that tell us? Well, it seems to tell us that the first 2 bases are very important for telling us which amino acid it's going to be, whereas the last base does not seem as important. It could be an A, it could be a C, it could be a G, it could be a U. So how do we explain this? Well, one, I can tell you that there are only 40 types of tRNA, which seems wrong, right? We have 20 amino acids. We have 61 different codons. Why don't we have 61 tRNAs? And I'll show you in a second why that is, but it's explained by this idea of the wobble hypothesis. The first 2 nucleotides are important to the anticodon base pairing, but the last nucleotide appears not to be. Now, this is going to be helpful for efficiency purposes. The dissociation of tRNA from mRNA is going to be able to produce faster protein synthesis, but I want you to understand why we might not need quite as many tRNAs because of this hypothesis. And so if we look at this image, I think it does a really good job. So you have your first base that really can't change because that seems important to determining the amino acid. The second base also can't change, but the third one could. So this is called the wobble base or the wobble position. So if G can base pair with C, which is what we would expect, cool, it's going to bring the correct amino acid. If it can pair with U, which is not what we'd expect, but still bring the same amino acid, that means that I can reduce the number of tRNAs that I need. Instead of having an AGA, which would perfectly pair with a U, I can get away with AGG and have one fewer tRNAs. So this is the idea behind the wobble hypothesis, and I think it's pretty cool. Now, putting it all together, I want you to think about all of the steps. So first, we had translation initiation that we basically just skip. We don't talk about it in this course, but understand that there's a lot of complexities there. Now, we also have to understand the roles of the three RNAs before we even start talking about elongation. So we have mRNA, which is serving as the message. That is what we see here in red. We are going to have rRNA in the ribosome itself, catalyzing the peptide bond formation between these amino acids to make the polypeptide. Then we have tRNA, which is bringing the appropriate amino acid to the ribosome at the A site and adding it to the growing chain at the P site. And remember that it was charged with the appropriate amino acid using the enzyme aminoacyl tRNA synthetase, which takes a whole lot of energy. So translation altogether is a very energy intensive process. And what I finally get out of it is a protein or a polypeptide. Now, I would love to say that we're done, but in reality, we kind of just got to here, which is the primary structure. The primary structure of the protein, which is just essentially the sequence. But hopefully you remember all the way back from Unit 1 that there's actually some more that can happen to that protein that almost certainly does happen. So this primary structure is actually going to be realistically undergo secondary structures, which are alpha helices and beta pleated sheets. Then that is certainly going to probably fold on top of itself and create a tertiary structure. And then some proteins will form a quaternary structure if there are several subunits to that protein. And remember that a protein is only going to have its final function once it reaches its final structure. And so this is a really important concept. Yes, we got pretty far with translation, getting that primary structure made, but really it's not until at the end that we have a fully functioning protein. And you should understand that there are some proteins that are involved in that called chaperones that are really assisting in this process. Chaperone is getting written on the screen right now. It is absolutely a term you should know. Certainly can strengthen any FRQ that I could give you. Much like a chaperone on a field trip sort of supervises the children to ensure that they're not getting into any trouble, a chaperone protein will ensure that the protein is being folded correctly. It's supervising the folding process. Now, we have talked a whole lot about how you get a fully functional protein, but not what happens really after you get that protein. And you have to remember that for the most part, my proteins are going to be highly regulated. And so I make modifications to this protein to regulate it. In terms of ones that we've already talked about, we've discussed phosphorylation. Remember, phosphorylation, you can either turn on or off a protein by adding phosphate groups to it. We talked briefly about glycosylation back in Unit 2, but there's also plenty of other post-translational modifications. Now, I also might not need a protein forever, and so my body might degrade it or my cells might degrade it, and they do that through a process called ubiquitination. Ubiquitination is a molecular tag that is added to proteins to sort of mark it for degradation, which will be degraded in a proteasome, which is lined with proteases, and then it will spit out a degraded protein, and those amino acids can be used again. Now, I want you to think about which one is going to probably be prioritized for most organisms, or at least most eukaryotic organisms. It's usually modifications. It's usually a smarter investment to just modify a protein and turn it on or off, rather than break it down and then remake it when I need it again. And that really comes down to the energy investment that is made during translation. There's a lot of energy that is used in translation, so if I can avoid having to make that protein again, that's typically prioritized energetically. That does not mean we don't degrade proteins. We absolutely do. All organisms do. But understand that that is typically not the more energetically favorable route. But you should know both of these. You should certainly know phosphorylation. You should certainly know ubiquitination. With all that said, that is translation and a little bit of post-translation regulation. And I'll leave you there. Have a good one.